before we get started today, I need to mention, as Cam said, that I am a co-founder and the Chief Innovation Officer of Psychedelics Therapeutics. But I thought I'd start today by talking a little bit about this new role at UC Davis that I've taken on as Director of the Institute for Psychedelics and Neurotherapeutics, or the IPN as we're calling this. And so, you know, the IPN is really a collaboration between the College of Letters and Science and the School of Medicine. And our ultimate goal is to achieve a mechanistic understanding of how psychedelics impact brain function and to use that information to develop better therapeutics, more efficacious, safer, more scalable, et cetera. So at the Institute, you know, we're interested in developing a lot of different types of therapeutics for brain conditions, but today I'm going to focus on the so-called stress-related neuropsychiatric diseases. These are conditions like depression, post-traumatic stress disorder, and substance use disorder. And a hallmark of all of these illnesses is really the atrophy of particular neurons in the brain, these cortical neurons in the prefrontal cortex. Now, the prefrontal cortex projects to a whole bunch of subcortical regions that regulate things like fear and motivation and reward. So it's perhaps unsurprising that these neurons would be impacted in these various brain conditions. Now, the current treatments that we have for these illnesses are far from ideal. These are molecules that have attempted to rectify chemical imbalances in the brain through the use of things like selective serotonin reuptake inhibitors. And these types of traditional antidepressants have some issues associated with them. First, it takes weeks to months for patients to start to feel any benefit from these types of medicines, and about a third of all patients will never respond to things like an SSRI. And so the approach that we're taking is a little different. We are trying to use small molecule drugs to specifically modulate select circuits in the brain. And the approach that we are taking really relies on physically changing the structure of neurons within those circuits in order to achieve long-lasting therapeutic responses. And so we started studying a class of molecules that we call psychoplastogens. These are chemical stimuli that produce structural neuroplasticity in the brain, such as the growth of these new dendrites. And if you zoom in on one of these dendritic these dendritic branches, you'll see these little tiny nubs called dendritic spines. Dendritic spines are the sites of excitatory synapses in the brain. And so these changes in structural plasticity often manifest as functional changes that you can detect with techniques like electrophysiology. Now, one of the first psychoplastogens identified, and perhaps the best known at this point, is this molecule on the left, ketamine. It's a dissociative anesthetic, been used in medicine for many, many years. But as Helen mentioned, this was recently approved by the FDA for treatment-resistant depression. And one of the theories as to how ketamine produces antidepressant responses has to do with its effects on structural plasticity. So here, I'm just showing you some cultured cortical neurons treated with a vehicle control or with ketamine. And you can see that the ketamine-treated neurons have more of these dendritic branches. And on the bottom here, these green dots are representing synapses. And so you can see that ketamine promotes both neuritogenesis, but also synaptogenesis. Now, here's some work from the late Ron Duman's lab and George Agajanian. And what they had demonstrated many years ago is that chronic stress will lead to the loss of those dendritic spines, which ultimately manifests as synaptic dysfunction. But what is really remarkable is that a single administration of ketamine can rapidly regrow those lost dendritic spines and reestablish synaptic connectivity in the cortex. And so when I started my program at UC Davis back in 2015, we were really interested in trying to find molecules that produce ketamine-like effects on neuronal structure, but maybe had some advantages. So ketamine's primary target is the NMDA receptor, which is found broadly across the entire brain. And so we thought very deeply about the genetics of the brain, and we're trying to identify targets that were selectively expressed exactly in the circuits that we cared about. And that really led us to study the serotonin or the 5-HT2A receptor. This receptor is highly expressed on those layer 5 pyramidal neurons that tend to atrophy in response to chronic stress. And you can see in the rodent brain, it's highly expressed in the anterior parts of the brain, with less expression as you move towards the back of the brain, and much lower expression in structures like the hippocampus or the striatum. Now, if you're interested in modulating the 5-HT2A receptor, one of the first classes of small molecules that you think about are the classic serotonergic psychedelics, molecules like LSD and psilocin, which comes from magic mushrooms. So one of the very first experiments that we did is we just took some cultured cortical neurons and we treated them with psychedelics. And what you can see is that compared to the vehicle-treated neurons in the middle, the neurons treated with the psychedelics DMT and LSD have substantially more complex dendritic arbors. And if you zoom in and do some super resolution microscopy on one of those dendrites, you can see that they also promote the growth of those dendritic spines and these philopodia-like structures. So now I'm just going to take a quick minute to explain one of the ways that we can actually quantify dendritic arbor complexity. We use something called shoal analysis. It's a very simple technique. All we do is we draw these concentric circles of increasing radii starting at the soma, and we simply count the number of times one of these branches crosses one of these circles. And that allows you to produce these really beautiful shoal plots like the one here on the right, where on the x-axis you have the radii of those concentric circles, and on the y-axis you have the number of dendritic crossings. Now, typically the way we represent the data is just looking at the maximum number of crossings of these shoal plots. So for something like the vehicle control here, that would be 7. And for something like DMT, that would be about 11. And what we were able to demonstrate is that psychedelics from 3 structurally distinct chemical classes, ergolines like LSD, tryptamines like DMT, and amphetamines like DOI, they're all really good at promoting cortical neuron growth. Now, to demonstrate this in vivo, we relied on a psychedelic that has a very, very short half-life in the body, dimethyltryptamine. The half-life of DMT in a rat is only about 10 minutes. And so for this study, we gave a single IP injection of DMT. We waited 24 hours, a time period when all of the drug has been cleared from the system. We then sacrificed the animal, took tissue, looked at neuronal structure via Golgi staining and function via electrophysiology. And what we find is that even after the drug has been cleared from the body, we see this very large increase in dendritic spine density in the prefrontal cortex. And this change in neuronal structure is accompanied by functional effects, including increases in both the amplitude and frequency of these spontaneous excitatory postsynaptic currents. Now, since we published this work in 2018, it has been replicated and extended by a number of groups throughout the world. But I just want to highlight one study in particular that was done by Alex Kwan's group back when he was at Yale. He's now at Cornell. And Alex was really interested in trying to understand just how long do these effects on structural plasticity last for. And so he took male and female mice, gave a single administration of psilocybin, and found that dendritic spine density was increased for at least a month after a single dose, which I think is a really, really remarkable result. Now, in addition to the rapid effects of psychedelics and their sustained effects after a single administration, psychedelics have another really interesting property, and that is that they have this panacea-like quality. They tend to work across indications. And I want to emphasize that we don't think these drugs are panaceas, but we do think that they might be targeting some common neural circuitry shared by many of these disorders, and that really involves the prefrontal cortex. We know from opto and chemogenetic revolution, some of the circuit level neurobiology that Cam was talking about in the introduction, that the PFC is a critical hub in the brain. It projects to a whole bunch of subcortical regions that do things like regulate drug-seeking behavior, modulate fear responses, and control motivation. And so I'm going to summarize a lot of work very, very quickly and say that in general, psychoplastogens, including psychedelics, tend to produce behavioral responses in assays that are known to involve PFC enhancement of fear extinction learning, which is relevant to the treatment of post-traumatic stress disorder, and also the production of antidepressant-like effects in assays like the forced swim test. Now, our group has been very interested in trying to understand the mechanism by which psychedelics promote cortical neuron growth. And so we started by just doing some selective pharmacology. We wanted to block certain receptors and kinases. We know that the 5-HT2A receptor is essential for the hallucinogenic effects of psychedelics. So one of the first things that we did is to block the 5-HT2A receptor with an antagonist. And when we do that, you can see psychedelic-induced effects on cortical neuron growth are completely abolished. We also wanted to block the effects of a couple of downstream kinases that have been implicated in ketamine-induced neuroplasticity. This one here, track B, is the high affinity receptor for brain-derived neurotrophic factor. And this one, mTOR, is a really critical downstream kinase that is involved in the production of all of the proteins that you need for plasticity, the structural proteins that allow neurons to change their shape, and the ion channels that allow them to change their synaptic weights. And if you block track B or mTOR, you completely block the effects of psychedelics. Now, at this point, we're just using antagonists, and the antagonist that we use for the 5-HT2A receptor was not a selective antagonist. It hits a few other receptors. So to be really, really sure that the 5-HT2A receptor was critical, we relied on genetics. And so here we were using these 5-HT2A knockout animals. And you can see in wild type animals, a single dose of 5-methoxydimethyltryptamine causes a very robust increase in dendritic spine density, but this effect is completely gone in the knockout animals. We're not the only group to observe this. Javier Gonzalez-Maizo's group has also demonstrated this using the psychedelic DOI. Now, in addition to the effects on structural plasticity, the functional effects are also gone in the 5-HT2A knockout animals. Again, wild type, you see an increase in both the frequency and the amplitude of these spontaneous excitatory postsynaptic currents, completely gone in the 5-HT2A knockout animals. Now, to assess the effects of the 5-HT2A receptor on the therapeutic behavioral effects of psychedelics, we ran this assay. This is a version of the forced swim test that is a little more challenging, where traditional antidepressants like fluoxetine tend not to produce responses, but these next generation psychoplastogens like ketamine do produce effects. So for this, we run a pretest. The next day, we give a single injection of the drug, measure head twitch response behavior just to make sure that it's impacting the targets that we think it is. And then the following day, we run another forced swim test. And what you can see here is that on the post-drug day, the 5-methoxy-DMT treated animals have this antidepressant-like response, but the ones that were pre-treated with ketanserin, the effect is completely blocked. Now, in addition to looking at these kind of motivated behaviors in the forced swim test, we also wanted to look at anhedonia. And for this, we relied on the sucrose preference test. Mice, much like myself, will prefer a sugary drink versus plain old water. And so we can measure this preference. And in wild type animals, you can see that there's a very robust sucrose preference, as is the same with the 5-HT2A knockout animals. But then after 10 days of chronic corticosteroid administration, a stressor, we can then do another sucrose preference test, and we can see a reduction in the sucrose preference in both genotypes, suggesting that there is this anhedonia effect. And then on day 13, we give a single dose of psilocybin, come back the following day and look at sucrose preference again. And we can see that that single dose of psilocybin can rescue the anhedonia effect in the wild type animals, but it cannot do so in the 5-HT2A knockout mice, really suggesting that the 5-HT2A receptor is really critical, not only for the hallucinogenic effects of psychedelics, but also for their effects on neuroplasticity and some of their therapeutic behavioral effects. Now, at this point, we had one result that was really strange to us, and it had us scratching our heads for a long time. And that was that this molecule right here, serotonin, was not able to produce the same types of effect on cortical neuron structure as a psychedelic. Now, that bothered us because serotonin is a very good agonist for the serotonin 2A receptor. And I just told you that the serotonin 2A receptor is essential for psychedelic-induced neuroplasticity. And so we didn't really understand this until we did this structure-activity relationship study. So basically what we were doing is we were taking serotonin-like molecules, modifying their structures a little bit, and seeing how that impacted their function. And what we found is that as we added these methyl groups, going from one methyl group to two methyl groups, we saw this really nice increase in psychoplastogenic effects. And so if you just look at these show plots, as you go from this very polar compound tryptamine to the more nonpolar N-methyltryptamine to the very nonpolar NN-dimethyltryptamine, you saw this increase in structural neuroplasticity. And when we just looked at activation of the 5-HT2A receptor in a HEK cell, we saw that activation of the receptor did not correlate very well with effects on structural neuroplasticity, but the lipophilicity of the compounds did. And so here you can see these very polar compounds like serotonin are not very good at promoting cortical neuron growth, but the very non-polar methylated compounds like DMT, they were much better at promoting cortical neuron growth. And that led us to the hypothesis that maybe the reason that serotonin cannot promote cortical neuron growth is because it can't access the target. Maybe the target is not on the plasma membrane, as is the case for most G protein coupled receptors. Maybe it's on the inside of the cell, and serotonin is simply too polar to passively diffuse across the nonpolar plasma membrane. And so to test this hypothesis, one of the first things that we did is just simply look at where the receptor is located. And so for this experiment, we took HEK cells, expressed 5-HT2A receptors and the beta-2 adrenergic receptor in HEK cells, as well as the CFP control. And you can see that these two G protein coupled receptors are expressed exactly where you would expect them to be. You have a high amount of receptor found on the cell surface. But what we found when we put this into cortical neurons is something very different. The beta-2 adrenergic receptor still had lots on the cell surface, but that wasn't the case with the 5-HT2A receptor. There was substantially more receptor localized intracellularly. And we're not the first to identify this phenomenon. There's several other groups have noticed this many, many decades ago, but no one really understood what the functional relevance of this intracellular population really was. And so to test the hypothesis that the intracellular pool of 5-HT2A receptors was the one that was important for psychedelic-induced neuroplasticity, we created a few chemical tools. The first set is we took these membrane permeable agonists, DMT and psilocin in green, as well as this membrane permeable antagonist, ketanserin in green, and we just converted them into membrane impermeable versions by making them permanently charged. So we have two membrane impermeable agonists and a membrane impermeable antagonist in purple. And then we ran the studies under two different conditions. The first is just normal cortical neurons, and the second condition is under electroporation. So with electroporation, you can apply a transient voltage that will open pores in the membrane, that will allow membrane impermeable compounds to cross the membrane, the pores close, and then they get stuck on the inside of the cells. And what we see is pretty interesting. Without electroporation, you get the results that you would expect. The membrane permeable agonist DMT and psilocin promote cortical neuron growth. The impermeable agonist TMT and psilocybin do not. Similarly, the membrane permeable antagonist can block DMT-induced cortical neuron growth, but the membrane impermeable antagonist cannot. But when you electroporate the cells and you can get those impermeable compounds on the inside, now you see that TMT and psilocybin can promote cortical neuron growth, and the methylated ketanserin can also block the effects of DMT. Now, this is looking at neurodegenesis. We ran a very similar experiment looking at spinogenesis, and we see the same thing. The membrane permeable compounds can promote spinogenesis. The membrane impermeable compounds cannot. Now, to really demonstrate that there are two populations of the 5-HT2A receptor. We relied on a fluorescent biosensor that is based on the 5-HT2A receptor. And you can see that in these cells, there's some on the cell surface and there's some inside the cell. And what we did is we pre-treated cells with the vehicle, and then we gave serotonin, or basically the fully methylated version of serotonin. You can think of 5-MeO-DMT as a greasy version of serotonin. And we can see that serotonin turns on the biosensor very nicely. 5-MeO-DMT a little less so because it's a partial agonist. But the next experiment was the critical one. We pre-treated these cells with a membrane impermeable antagonist. So this can only block the receptors on the cell surface. And when we do that, we see that all of the signal coming from serotonin is gone. But the signal coming from DMT is not completely blocked. There's still some residual signal suggesting that 5-MeO-DMT can access this intracellular pool of receptors. Now, at this point, we reasoned that if it really was all about the location of the receptor and that serotonin just can't access the intracellular pool, if we could find a clever way to get serotonin inside the cell, it should be able to turn on the receptor and cause cortical neuron growth, just like psychedelics. And so for this experiment, we sparsely expressed a transporter for serotonin, SERT, on a subset of neurons within the culture. So here you can see two neurons, they're side by side. The one on the left expresses the transporter, the one on the right doesn't. And similarly over here, the one on the right expresses the transporter, the one on the left doesn't. That means that some of these neurons can import serotonin and some of them can't. And then what we find is that when we treat with the vehicle for either genotype, we see no effect. Dimethyltryptamine, the membrane permeable agonist, promotes growth in both genotypes, but serotonin only promotes growth in the cells that are capable of importing the monoamine. We see a very similar result for spinogenesis assays. Again, no effect of the vehicle. DMT promotes growth in both genotypes, but serotonin only promotes growth in cells that are capable of importing serotonin. So to demonstrate this in vivo, we relied on using a viral construct. So we used a virus to express the serotonin transporter in those neurons in the prefrontal cortex. Now, they don't normally express this transporter, so normally serotonin cannot get inside them, but with the transporter, now they can suck up serotonin. And after the transporter was expressed, after about 3 weeks, we gave a single dose of parachloroamphetamine. This is a compound that causes the release of serotonin. And then 24 hours later, we look at spine density. And what we find is that only in the cortical neurons that can import serotonin do you get this increase in dendritic spine density in response to a serotonin releasing agent. And we see similar effects on behavior. So again, we express the transporter. We then run a novelty-induced locomotion assay just to make sure there's no difference between the genotypes. We run a pretest for the forced swim test, and we see again the genotypes are identical. We then give parachloroamphetamine. 24 hours later, come back, and what we see is we get an antidepressant response only in the animals that have the ability to import serotonin into those prefrontal cortical neurons. Now, at this point, we are pretty convinced that the serotonin 2A receptor is critical for the neuroplasticity-promoting effects of psychedelics and some of their antidepressant-like effects. But we do know that the serotonin 2A receptor exhibits a wide range of, quote unquote, functional selectivities. And there are activators of this receptor that are non-hallucinogenic. And so a big question that we had was whether or not we could separate the hallucinogenic effects of the drugs from their beneficial effects on neuroplasticity and their antidepressant responses. And I know Cam said it was my life goal to take the fun out of fungi. I don't think that's my life goal at all. I think that there's going to be a subset of patients who are definitely going to be helped by these first generation psychedelic medicines. But there's some issues associated with that type of treatment, namely one of scalability. Right now, in order to administer them safely, they have to be given under the supervision of a healthcare professional, which drastically increases the cost and complexity of the treatment. And so we reasoned that if it was possible to get some therapeutic effect from these non-hallucinogenic variants, we might have more scalable treatments to reach a larger patient population. And what is really important to consider is that 1 out of 5 people will suffer from a neuropsychiatric disease at some point in their lifetime. We're talking about 1 billion people worldwide. And so we are in desperate need of scalable solutions, take-home therapeutics to help all of these different patients. So in order to achieve this goal of trying to identify a non-hallucinogenic analog of a psychedelic with therapeutic effects, we needed a really good assay that could predict hallucinogenic effects in people. And for that, we turned to this behavioral assay called the mouse head twitch response. This is a paper published by Adam Halberstadt's group, really beautiful work demonstrating that across over 30 structurally distinct psychedelic compounds, there is this exceptional correlation between potency in the head twitch response assay and human hallucinogenic potency. And so for a lot of our work, we'll rely on this rodent assay, but it's not really amenable to drug discovery. You can't screen thousands of compounds in a behavioral assay like this. It's just not, it's not practical. And so we really needed a cellular assay to predict hallucinogenic potential. And so the way we did this was to take the serotonin 2A receptor, and we simply lopped off the third intracellular loop of this GPCR, and we fused on a circularly permuted green fluorescent protein. And the way these CPGFPs work is the beta barrel is essentially split and it's open, which allows water to get in to quench the fluorophore. But when a ligand binds to the receptor, that induces a conformational change that closes the beta barrel and allows fluorescence to turn on. So you can see a nice concentration response with serotonin here. And you can see that the antagonists, ketanserin and MDL, essentially do nothing or they turn off the sensor. And so we got very excited when we found that human hallucinogenic potency correlated very nicely with potency in this biosensor assay. So we started calling it Psychlite. And one of the first experiments that we did is we used a virus to express Psychlite in the prefrontal cortex. We then gave a single administration of 5-methoxy-DMT. And what you can see is that Psychlite fluorescence turns on very rapidly. And this correlates very nicely with the onset of the head twitch response behavior that I just told you about. What is very interesting, though, is that Psychlite fluorescence seems to stay activated for some time while the head twitch behavioral response declines. Now, if you give a vehicle injection, you see no change in Psychlite fluorescence. And if you give an antagonist, you actually see a slight decrease in Psychlite fluorescence. Now, I would say probably the day that I was most excited was when we saw these data. And we wanted to see if Psychlite could differentiate between a hallucinogen and a non-hallucinogenic congener. And so here I'm showing the hallucinogenic molecules LSD and 5-methoxy-DMT, and I'm showing non-hallucinogenic analogs glyceride and 6-methoxy-DMT. Now, glyceride is almost identical in structure to LSD. The only difference is you flip that stereocenter and add a nitrogen. For 5-methoxy-DMT, if you just move the methoxy group one position down on the indole, that's how you get to 6-methoxy-DMT. Now, what you'll notice is that the hallucinogenic ligands in green both turn on the sensor very, very nicely, but the non-hallucinogenic ligands do not. Now, you might be asking yourself, well, maybe the non-hallucinogenic compounds don't bind to the biosensor. So to address that question, we can simply run the assay in antagonist mode. So we give a little bit of agonist to turn on the sensor, and then we titrate in increasing quantities of the non-hallucinogenic compound, and you can see the fluorescence decrease. And that's exactly what we see in the graphs here on the right. So next, we simply created a stable cell line that expresses the Psychlite construct. We call these Silly-2 cells. Silly-2 cells are quite nice because they have some baseline fluorescence, which allows you to detect inverse agonism as a decrease in fluorescence intensity and agonism as an increase. Now we can run the assay mode on the x-axis and antagonist mode on the y-axis. And I just want to point your eye to this black dot right here. This is the vehicle control. And what we found was that compounds that increase psycholyte fluorescence in agonist mode more than 1 standard deviation from the vehicle, these are all of these known hallucinogenic compounds in red. Compounds that decrease psycholyte fluorescence more than 1 standard deviation from the vehicle in antagonist mode, these are all these non-hallucinogenic ligands in blue. Now we could simply take agonist and antagonist data and combine them to make this ligand score that we have on the left here. So molecules with a red ligand score tend to be hallucinogenic. Molecules with a black ligand score tend to not bind to the receptor at all. And molecules in blue tend to be non-hallucinogenic ligands of the serotonin 2A receptor. And if you look at this list, you can find some really interesting things. You'll see traditional antagonists like MDL and ketanserin. You'll see antipsychotic compounds like haloperidol, and you'll even see some of these non-hallucinogenic agonists like glyceride. So I want to briefly tell you about 2 of these compounds. I want to start with this 5-bromo-DMT compound. We were really interested in this because we had made the entire 5-halo series going from fluorine to chlorine to bromine. And really all we're doing here is just increasing the van der Waals radii of those atoms. And what we found was that based on the psycholyte biosensor, the fluoro and chloro compounds were predicted to be hallucinogenic, but the bromo compound was predicted to be non-hallucinogenic. And that's exactly what we see in the head twitch response. The fluoro compound produces robust head twitches at all doses. We get this nice dose-dependent increase in the chloro compound, and the bromo compound doesn't produce head twitches at any dose tested. And similarly, we found this compound here that we call AAZ, A154, named after the student who synthesized it, but it's kind of structurally related to 5-MeO-DMT. You can squint your eyes and kind of see how they're related. And what the biosensor indicated was that AAZ should be non-hallucinogenic. We don't see any head twitch response, even up to these massive 100 mg per kg doses, but a therapeutic dose is a lot closer to about 10 mg per kg. Now, what's really interesting is the AAZ, even though it's non-hallucinogenic, has really potent psychoplastigenic effects. This is just showing that it's comparable to ketamine in that respect. It also has long-lasting and rapid antidepressant effects. So this is a case where we looked at forced swim test behavior, compared it to ketamine. After 30 minutes, we see a rapid antidepressant effect, and we also see a sustained antidepressant effect that lasts for at least about a week in this assay. And then this is an assay looking at anhedonia using these VMAT2 heterozygous mice developed by Bill Wetzel's group at Duke. And for this, you can see that the wild type animals have this really robust sucrose preference, but these are gone in the VMAT2 heterozygous mice. We know that in humans, if you inhibit VMAT2, you can induce depressive phenotypes, and these mice have this baseline anhedonia phenotype. We give a single dose of AAZ, and we find a rapid rescue of this anhedonia, and it seems to last for at least a couple of weeks before the genotypes really start to diverge. Okay, so we've been able to use Psychlight to identify a whole bunch of these non-hallucinogenic psychoplastogens that might have uses for various indications. But I want to talk about one right now based on this molecule, ibogaine. Now, ibogaine is not a classic psychedelic. Maybe you would call it an atypical psychedelic, but it has some really interesting properties. There are some anecdotal reports and open label clinical trials that suggest a single administration of ibogaine can keep heroin addicts drug-free for up to 6 months, and a second dose can keep them drug-free for up to 3 years. And that is really, really remarkable and just screams effects on neuroplasticity to me. So you might be asking yourself, well, if ibogaine is so effective for treating substance use disorders, why are we not using it to address the opioid epidemic in this country? And the reason for that is that ibogaine is a terrible drug for a variety of reasons. Number one, it's really hard to synthesize. There are no de novo total syntheses that can produce it enantioselectively in the quantities that we need for clinical trials. and getting it from the plant sources has a whole bunch of ecological issues and other problems associated with it. Number two, if you've ever worked with ibogaine, you know it's a rock. It's completely insoluble in water, which makes formulation incredibly challenging. Of course, it has hallucinogenic effects, but probably the worst thing about ibogaine is that it binds to an ion channel in the heart called hERG, which can induce cardiac arrhythmias. And several people have actually died during their ibogaine treatments for their substance use disorders. And so our approach to this was pretty simple. It's something called function-oriented synthesis that's really popularized by Paul Wender at Stanford. And the idea is very simple. The idea is, can you take a complex natural product like ibogaine and distill it down into the structural features that are essential for the function that you care about? And so in the case of ibogaine, there's really three key structural motifs. There's this indole group, this 7-member tetrahydroazepine ring, and this bicyclic isoquinoline. And so all we did is we started deleting bonds. And so if you delete the bond between the indole and the isoquinoline, you make molecules that look like scaffold 1. If you delete the isoquinoline altogether, you make molecules that look like scaffold 2. I'm not going to take you through all the chemistry. I'm just going to highlight a couple of things. Basically, what we found is that molecules that retained the azepine but lacked the isoquinoline, they were pretty good at promoting cortical neuron growth, but the opposite wasn't true. Compounds that contained the isoquinoline but lacked the azepine, well, they were pretty bad for the most part. So we decided to prioritize this compound for further study. We started calling it iboganolog, or IBG, because it resembled the natural product ibogaine. We made one more small structural tweak. We just moved the methoxy group from the 5 to the 6 position. Now we started calling this compound tabernanthalog, or TBG, because it resembled the natural product tabernantine. Now, unlike ibogaine, we can synthesize tabernanthalog in gram quantities in a single step using this Fischer-Indole cyclization. Both IBG and TBG are very, very water soluble, and they're both potent agonists of the 5-HT2A receptor. Now, I want to show you this one really interesting structure-activity relationship study. You'll notice that the structure of 5-methoxy-DMT is embedded within the ibogaine framework, and you can see that it produces a very robust head-twitch response. But if you conformationally restrict the dimethylamino group to make IBG, you can drastically reduce that head-twitch response and move the methoxy group one position over. Now you've got a completely non-hallucinogenic analog. Now, in addition to being non-hallucinogenic, TBG has a significantly safer cardiotoxicity profile. It is about 150-fold less potent on hERG than ibogaine. And you can actually visualize this looking at these immobilized zebrafish. This is the heartbeat from the vehicle-treated fish. You can see the bradycardia induced by ibogaine, and that's not there in the TBG-treated fish. Now, as a measure of propensity for inducing cardiac arrhythmias, you can simply take a ratio of the atrium to ventricle beats per minute, and you can see that ibogaine is very good at promoting arrhythmias, as is the positive control Sir-Indole, but you can see that TBG looks just like the vehicle control. Now, in addition to having cardiotoxicity issues, ibogaine has some other just general toxicity issues. You can look at these graphs here on the left, but I think a picture is worth 1,000 words. This is what a vehicle-treated fish looks like. This is what an ibogaine-treated fish looks like. They're very, very different, but a TBG-treated fish looks more like the vehicle control. Now, it's great that we created a safer version of ibogaine, but none of this really matters if it's not going to be efficacious. So we wanted to confirm that it was a very good psychoplastogen. And so for this experiment, we relied on mice that expressed a fluorescent protein in the dendrites of those cortical neurons in the cortex. And that allows us to visualize those dendritic spines on the first day. We then give a single dose of TBG, come back the next day, 24 hours later, when the drug has been cleared from the system, and we look at the exact same section of dendrite. And that allows us to quantify how many dendritic spines were formed, like this one here in blue, and how many were eliminated, like the one here in red. And what we find is that TBG does not really impact spine elimination, but it does promote spine formation, and it does so to a comparable extent as this hallucinogenic drug, DOI. Now, if our hypothesis is correct that promoting structural plasticity in the cortex is important, we should see broad therapeutic effects of TBG, and that's exactly what we see. We see antidepressant-like effects in the forced swim test. We also see a really profound reduction in alcohol consumption in this 2-bottle choice paradigm. And then finally, we looked at heroin use disorder, and we found that no matter if you administered TBG during self-administration, during the early stages of extinction, or immediately prior to cued reinstatement, you had this really long-lasting protective effect on heroin-seeking behavior. And then finally, the last thing I want to emphasize is that TBG and psychoplastogens in general have the ability to heal damaged neural circuits. And they're really taking neuropsychiatry in a direction away from just treating disease symptoms and trying to actually heal damaged circuitry. And so what I'm showing you here is some calcium imaging in the somatosensory cortex. Now, mice are very tactile creatures. They like to explore their environment with their whiskers. And every time that they whisk, you'll see this really nice coordinated calcium response in the somatosensory cortex. But unpredictable mild stress can completely disrupt this coordinated response. But what is really remarkable is that a single administration of TBG, long after the drug has been cleared from the body, can restore this coordinated response. And so to summarize what I tried to tell you about today is that in a healthy brain, the neurons in your cortex have lots of branches and lots of dendritic spines so they can communicate with their neighbors. But in many brain disorders, including the stress-related neuropsychiatric diseases like depression, you see a frank atrophying of these neurons. In psychoplastogens, including hallucinogenic compounds, as well as non-hallucinogenic compounds, are very good at promoting the growth of these cortical neurons. So in summary, I told you that psychedelics promote structural and functional neuroplasticity primarily through the activation of this intracellular population of 5-HT2A receptors. That Psychlite is a very good biosensor for predicting hallucinogenic potential, and that TBG and AAZ are two potentially safer, more scalable psychoplastogens. And I'd like to thank all of the funding agencies on the right who contributed to this work, and all of the really key collaborators, both from within UC Davis and outside of UC Davis, that played important roles in this work. In particular, I want to thank John Gray. His lab did all the electrophysiology that you saw in this presentation. And John is the associate director for the IPN. And it's been incredible working with him trying to build this institute. And I'd also like to thank Lynn Tien. It was a really fantastic collaboration working with her group on the development of Psychlite. And finally, I need to thank all of the incredibly creative, hardworking, and just overall talented individuals I get to work with every day, the postdocs, grad students, and undergraduates who really drive the science. Here, I'm showing you in blue former students whose work I talked about today, and in white are all of the current students in the group, and I'm really, really excited to see where they take the science in the future. And with that, I just want to thank all of you for your attention, and I'd be happy to answer any questions.
I think we have about 5 minutes for questions, David. I'm going to read you a question from our online folks, and it is a long question, but it's a really good question. So if mTOR inhibition decreases psychedelic-induced neuroplasticity, then it is likely that metabolic therapies like fasting and ketogenic diets may also decrease the beneficial effects of psychedelics on neuroplasticity, associated mood and cognitive functioning. This is relevant to cancer patients undergoing chemotherapy or radiotherapy, where adjuvant metabolic therapies are used to reduce treatment toxicity and optimize anti-cancer effects, and who are often also undergoing mood-targeted treatments, which may include psychedelic-assisted therapy.
So the way I'll answer this is that right now, very little is known about what compounds a patient can and cannot be on to still get an effective psychedelic. And if you are using modulators of mTOR, you can imagine that might be an issue. But you have to consider if the compound is going to actually cross the blood-brain barrier. So for instance, there's an example where Ron Duman's lab and John Crystal and others, they were using the compound rapamycin, which inhibits mTOR and will block the effects of ketamine and psychedelics on structural plasticity in culture and in rodents if you directly inject it into the brain. But if you give that to a human, they actually found that it extended the effects of ketamine, and that was pretty surprising to them. But there's not a lot of evidence that suggests that that compound can easily cross the blood-brain barrier. It's very, very large. And so maybe what is happening is that we're getting inhibition of mTOR in the periphery, and that's having some immune modulatory effects that might be interacting with the plasticity effects that you're getting in the central nervous system. And so that's something to always keep in mind. Now, in terms of other things like fasting, other diets, that I think is still a completely unexplored area for psychedelic medicine.
Hello, I'm super excited, Dr. Olson, because I have used your cell report in a number of my graduate papers. So I'm super happy to be here. And one of the questions that I work in psychedelic therapy now out of a company in Miami. I work remote. Some of the things that I've noticed among the patients that I help is that some of them are even resistant to the ketamine treatment. My theory is that it's due to a number of things. 1, set, 2, setting, and 3, the willingness to reflect. The other component is that, like, how do we assess chemical psychosis, chemical imbalance versus a spiritual crisis? Because I believe that the patients who are undergoing spiritual crisis can benefit greatly from psychedelic therapy, while those who may need more chemical balances may not, because I've seen that as well, where it just furthers their psychosis. So how do we determine the two, and is there a plan to do that?
Yeah, you're bringing up a really important point, and that is the idea of personalized medicine and patient stratification. How do we identify the patients that are likely to respond to one type of medicine versus another? And now I will tell you that there are some efforts to do this right now, thinking about the neuroplasticity aspect. There are imaging modalities that we can use to measure surrogates of synaptic connectivity, even in a human brain. And there is some indication that suggests that some patients have really severe cortical atrophy. They might be the ones that respond better to, a psychoplastogen, and other patients maybe have less severe cortical atrophy or no cortical atrophy, and that might be a different patient population. Now, actually, one of our speakers later today, Connor Liston, has done some really incredible work using imaging to stratify different subtypes of patient populations. And the idea is, if we can identify them early, we might be able to give them the appropriate treatment. And so therefore you can enhance the overall efficacy.
Can you hear me now, David?
There we go.
Okay, great. Putting your data together, since TBG is highly water soluble, how would it be possible for TBG to cross the neuronal membrane to act through intracellular 5-HT2A receptors?
Yeah, so this is the trick of good drug discovery. And I always say there are medicinal chemists and then there are CNS medicinal chemists. You have to play with a lot of factors. You've got to make sure that it's soluble in the blood. You also have to make sure that it's not going to be so lipophilic that it's just going to get stuck to proteins in the blood. There's a fine balance. So it turns out TBG actually has very, very good membrane penetration. It actually reaches extremely high concentrations in the brain, and that has to do with how we engineered it to have these specific physical properties.